Everyone for joining us tonight. We're approaching 30 participants. Thanks so much for our speaker tonight, Professor Björn Corzilius. We're very excited to have him here. He's working on sort of spin selective solid state DMP, like polarizing selective spins in molecules in the solid state. That is very interesting, I find. He's right now professor for physical chemistry in Rostock at the University of Rostock. Before that, he studied chemistry in Darmstadt, and where he did his PhD as well. And then he went to Bob Griffin's lab at MIT. Then he came back in the Minuta program. That's some history that we share. It's a great program I can recommend for everyone. The Minuta program of the German Science Foundation. And shortly after, he became appointed professor in Rostock, where he still is. So without much further ado, very excited to hear tonight. Björn, thanks for being with us. Everyone else, please, we'll have a discussion in the end, so keep your questions and we'll come to them. Thank you so much, everyone.
Well, thank you very much, Jan. And it's a pleasure to be here at the Kiel imaging seminar. So it's not so far away from Rostock, but with Zoom it's even closer practically. So I didn't even have to leave my office. Well, thank you for staying with us tonight, and so that I can tell you something about our research projects here in Rostock. So as you can see here, the title of my talk is Site-Specific Dynamic Nuclear Polarization in Biomolecular Magic Angle Spinning, NMR. So a quick disclaimer that we're not doing any imaging here, but as we are all interested, I think, in spin physics and spin interactions and how to make spins visible, either via imaging or via spectroscopy, I think we are still all interested in this. So let's come to the next slide. I think I have to activate my PowerPoint again. So here you can see the outline of my talk. And in the beginning, I will give a quick introduction to magic angle spinning dynamic nuclear polarization, because that might be something that you're not so familiar with. So I will show you first how DNP actually works, what's the concept behind it, what instrumentation we need, and so on. Then I will give you an introduction on a hammerhead ribozyme, how we can actually measure the DNP enhancement and benefit from the signal enhancement. And in the end, how we can actually use this to extract information about the fold of a biomolecule. Then the main part of my talk will be about heteronuclear cross-relaxation under DNP, a new method that we developed, Scream DNP, and how we can use that for in two examples, for an intra-complex transfer in a tetracycline binding aptamer, and to use site-specific magic angle spinning NMR in a ribonucleoprotein. And the final part, I won't have time today, but if you're still interested in that, I can give you a short glimpse probably in the discussion, how we can use direct DNP of low gamma nuclei such as carbon-13 or even nitrogen-15 in order to extract spin-spin distances between the electron spin and the nuclear spins. So let's come to DNP. And if there is one problem that NMR is suffering from, then this is usually sensitivity. And this is not only NMR, but also imaging. This is why sequences take a long time for us. Spectral recording takes a long time, so sometimes weeks or even months. And this is due to the relatively small Zeeman interaction, which gives us the resolution on the one hand, but also makes our lives more complicated. So if we think about this as physicists or physical chemists, then we know how to deal with absorption and or absorption spectroscopy. Of course, with NMR or imaging, we are not really measuring the absorption signal, but as an analogy, I think this makes it more intuitive to understand. So if we only consider our spins inside of the magnetic field to be in the energetic ground state, then we would have no problem at all. We would see a large absorption signal if we can excite them practically by spectroscopy. However, due to the small energy difference between the spin substates and the relatively large thermal energy, we have an almost equal population of the excited state. And this then leads to what we know in absorption spectroscopy as stimulated emission. And in the end, the two signals cancel each other, and we only are left with a very small net signal. We can quantify that by looking at the Boltzmann spin polarization, and we see that under typical conditions for NMR, we are left with only about 0.01% of spin polarization. We know that we can compensate for this relatively small signal that we measure. This number means just that if we have 10,000 spins in our ensemble, then only one of those 10,000 spins actually gives a measurable signal. And we can compensate for this small signal by averaging over more transients. But we also know that we measure more noise per transient, or with many more transients, we absolutely measure more noise, which scales with the square root of the number of scans. So our signal to noise ratio only grows with the square root of the number of scans here. And that means that there's actually a potential of 100 million-fold time savings in acquisition time. So this is what we have, what the physics practically provides to us. And even if we can only harvest practically a small fraction of this, we can still benefit from this tremendously. And this is what dynamic nuclear polarization does. It continuously transfers electron spin polarization to the surrounding nuclear spins. And this is because in comparison to a proton spin, the electron spin has a much larger Zeeman energy and therefore also a much larger spin polarization. And compared to a proton, this is about 660 folds larger. And if we do this, we can transfer this electron spin polarization to protons, for example, and also increase the NMR signal by a factor of 660-fold. This is what we call here the maximum NMR signal enhancement. This is a theoretical factor which is calculated directly from the gyromagnetic ratios. In experiment, we will measure a slightly smaller ratio because, of course, our transfer is not ideal. We have some losses, for example, due to relaxation or insufficient excitation. But still, we have measured something up to 250-fold experimental DNP enhancement factors. And this then is practically the NMR signal increase that we can gain. For this, what we need is special instrumentation. Here you can see the typical NMR magnet with the NMR probe. There's a magic angle spinning probe because we do everything in solid state. What we also need is a microwave source. And here you can see the scheme practically of a gyrotron. Gyrotron is a linear electron accelerator tube. And without going into detail how this works, in the interaction cavity, the microwaves are generated and then they are coupled here by a mode converter into an overmoded waveguide. And this then guides the microwaves into our sample chamber here, into the MAS stator, directly onto our rotor. And this excites practically the DNP transitions. What we also need is a cryogenic MAS system so that we can actually cool our system or our sample down to temperatures of around 100 Kelvin while doing MAS. So in our lab, we are using a 9.4 Tesla magnet, which corresponds to 400 megahertz proton frequency. I've mentioned the cryogenic MAS system. And our microwave frequency is then at 263 gigahertz, which is practically the EPR frequency at 9.4 Tesla. And what we have is a 3.2 and a 1.3 millimeter DNP probe. Here you can see the sample rotors. And the 1.3 can spin up to 40 kilohertz at 100 Kelvin. So here you can see our setup at the Department Life, Light and Matter. Here you can see the building of our research building in Rostock. And here in the basement, practically, there's our DNP lab. Here you can see the gyrotron with its own magnet and high voltage supply. And here all the cryogenics practically is operated and controlled for magic angle spinning. All right, I have to spend a few words on sample preparation because I've mentioned that we do everything in solid state, and this is why we need magic angle spinning, of course, to average the anisotropic interactions to get solution-like spectra. And we do this because we have to cool everything down to temperatures of about 100 Kelvin. And then we practically have a frozen amorphous solid consisting of our analyte, which is co-dissolved practically with a polarizing agent. And the solvent matrix is a mixture of glycerol, usually D2O and H2O. This is the typical so-called DNP juice, practically, that we use for all biomolecular samples. And we can vary practically the ratio between glycerol and D2O and H2O. And therefore also we can control our proton density practically in the sample. As polarizing agents, or the polarizing agent is the molecule practically that provides the electron spin polarization and then allows this to be transferred to the surrounding nuclei. We can use such tailored bis nitroxides, and they are specifically tailored for cross-effect DNP. I don't have the time to go too much into DNP mechanisms, but for the content of my talk, it's also not so important. And besides these bis nitroxides, we can also use other systems. For example, I'm showing here such gadolinium chelate molecules or complexes. This is gadolinium DOTA, and you might know that as DOTA-REM, for example, as a contrast agent. And we have introduced this more than 10 years ago as polarizing agent for magic angle spinning DNP. The advantage of this can be that we have more control over the polarization by using the solid effect, which is a simpler mechanism, but it's not as efficient as the cross effect with bis nitroxide. So everything I'm showing you today will be using amopol as a polarizing agent and using cross-effect DNP. And then under these conditions, we know that the electron spins in this amopol will transfer the polarization over the whole matrix uniformly in the proton bar. So all the protons in the sample will be uniformly hyperpolarized to the same enhancement factor practically. And with this, I just want to show you one example where we utilized DNP to investigate the fold practically of the hammerhead ribozyme. The hammerhead ribozyme is a catalytically active RNA molecule or a motif in most ubiquitous in the plant kingdom. And here it plays a role, for example, in plant viruses and viroids, where it senses a change in the divalent metal ion concentration upon infection of a host cell. And once this magnesium, for example, or manganese is present, it will it will trigger a catalytic acid-base reaction, which cleaves the RNA, also a hybridized DNA, and then releases the viroidal genome for replication. When this was investigated, there was a minimal structure which was created practically so that this could be investigated by solution NMR. However, it was found that the catalytic activity is reduced by about a factor of 10 as compared to the natural activity of the wild type, or the full length hammerhead. And then it took about a decade for Scott and Martic to hypothesize practically that there are interactions between two stem loops, which are crucial for pre-folding of the RNA, which then allows for a much faster catalytic cycle where the divalent metal ion just has to hop into the binding site, into the selective binding site. And then the catalytic activity can be triggered without the RNA have to fold first. And but it was not clear, is this really the case, or was the concurrence practically between the apo and the holo structure only a crystallization artifact? And this is where our research then kind of came into play. So we were asking, can we investigate this in frozen solution by NMR, by magic angle spinning NMR? And so the idea behind this is you freeze practically all the different conformation, which are dynamically interchanging in solution, and then you get an ensemble practically of all the different conformers, and you can investigate those by NMR. However, even with a uniformly labeled carbon-13 and N15 labeled hammerhead, full-length hammerhead RNA, we have not been able to get enough signal intensity for any more sophisticated experiments. But then we can do our DNP magic. Practically, we have Amopol in our solution. We just turn the microwaves on, and then we get this 250-fold NMR signal enhancement, which practically saves us more than 60,000 folds practically in time. And then we can do in a few minutes what would take more than a year without DNP, and we can practically get noise-free NMR spectra of carbon-13 and N15. But we still have a problem, and this problem is spectral crowding. So if we look here at this carbon-carbon correlation spectrum, and we consider that we have a large RNA of about 53 nucleotides, I think, then we come to the conclusion that there are almost 500 carbon-13 nuclei in such a molecule, and we expect on the order of 10,000 signals in such a 2D spectrum. And you can see that this means that every peak you see here is a convolution practically of dozens of single sites. So we have no way of disentangling this. And this becomes clear that because we have a limited number of different monomers which are involved in making up such a biopolymer. And so we lose usable information because we cannot extract it specifically. There are ways around this, and we've briefly talked about this already with Jan. Here you can see that we have used an isotope labeling strategy where we had a hammerhead ribozyme which is constituted in trans. That means we have two single strands which are then hybridized, and this allows us, for example, to use in vitro transcription to use only uridine labeling or only adenosine labeling in our substrate strand, you can see here. Or or orthogonally labeled enzyme strand, which you can see here, which is only N15 labeled, but this on all nucleotides. And then we break this down practically to only 4 nucleotides, which contribute to our signal. And we only see interstrand contacts practically between the substrate strand from the carbon-13 and the N15s on the enzyme strand in such a 15N carbon-13 correlation spectrum. And what we could do here is we could identify the unique signature of one reverse Hookstein base pair. And this reverse Hookstein base pair can only exist here between this uridine and this adenosine here. And this shows us clearly the existence of the stem-stem or loop-loop interaction between those 2 stem loops. And more importantly, in the apo hammerhead ribozyme without magnesium, there was practically the same fingerprint here. There's no qualitative or quantitative difference between the 2 spectra. So this clearly shows that the fold is preserved even in the complete absence of any divalent metal ion, which fully backs Scott's and Martig's hypothesis that the hammerhead ribozyme is pre-folded in solution, and only the divalent metal ion has to hop in practically and immediately is able to trigger the catalytic cleaving. All right, now for the last about 10 minutes, I will jump here to a new method that we have developed. This is heteronuclear cross-relaxation under DNP, and how we can use this to introduce site specificity for DNP in addition to isotope labeling strategies. What we have used with the in vitro transcription for RNA labeling, this is not site-specific labeling. This means that we can only, for example, add uridines or adenosines. But if we look at large complexes, then this will also then label all the nucleotides of one type practically without selectivity here. Because at some point, for example, solid phase synthesis for such biopolymers becomes tedious and very expensive. So how can we do or combine practically site specificity with simple biochemical isotope labeling strategies? And this is by making use of cross-relaxation, of heteronuclear cross-relaxation, which spontaneously happens under solid-state DNP conditions. And we have found this effect practically by more or less accident in a protein, where we, after, it probably took us about a year to really understand what's going on. But in the end, we figured out that the methyl groups in the protein, in the amino acids, are still dynamically active, even at temperatures of 100 Kelvin, which then leads to a modulation of the dipolar interactions between those protons and this carbon nucleus. And this then leads to cross-relaxation. So this is very similar to the heteronuclear Overhauser effect. And we can practically use the whole theory of this and explain everything. The only difference is that we do not saturate our proton spins during this experiment. So with a normal NOE experiment, you irradiate the protons, you saturate those, and you practically then wait for the cross-relaxation, because this drives them back to the thermal equilibrium and then kind of takes the carbon polarization with it. What we do is we hyperpolarize protons, so let's say 100-fold, which is a much larger deviation from thermal equilibrium. And then also we get a much stronger effect, because this scales then practically with the proton enhancement factor. But the underlying mechanism is practically the same. What's also noteworthy here is that this kind of comes with a signal inversion in the carbon-13 spectrum. You can see this here, that whenever we see this effect, we see not absorptive but emissive signals. And we can easily use this phase difference practically by 180 degrees to differentiate between the different mechanisms active. So is it a direct DNP of carbon-13? This gives us positive signals. Is this heteronuclear cross-relaxation? This gives us negative signals. Now we can separate these two contributions and practically get pure either direct polarization or pure heteronuclear cross-relaxation spectra by doing this interleaved acquisition with a saturation step on the protons here, and then a mathematical subtraction. And this whole set of experiments we call SCREAM DNP, which stands for specific cross-relaxation enhancement by active motions under DNP. And now let me show you first how we can use the different dynamics, for example, of different amino acids, and also use this as a measure for internal molecular dynamics practically. So all the aliphatic amino acid side chains here have very similar dynamics, and you can see that they also behave very similar with this respect here. In the orange spectra, you can see practically the heteronuclear cross-relaxation contribution, and these are also always in the range of about -10 to -20 enhancement. There are two methyl-bearing amino acids which kind of stick out. One is methionine, which gives a very large SCREAM DNP enhancement, and there's threonine, which gives a rather small SCREAM DNP enhancement and a much larger direct DNP enhancement on carbon-13. And this is because the methyl dynamics are varying or are differing tremendously between here the SC bond and here the CC bond in threonine. Also in threonine, you might have interference with the OH group, which furthermore slows down the methyl rotation. All right, but now I just want to show you how we can use this and what we can do with this. This is an example on the tetracycline binding aptamer. This was done in, whoops, this was wrong, sorry. Here we are. This was done in collaboration with Beatrix Süß from TU Darmstadt. And so the tetracycline binding aptamer is an artificial RNA which has been tailored practically to bind the antibiotic tetracycline with a very high affinity, the dissociation constant of only 0.8 nanomolar. However, it requires magnesium 2+ as an essential cofactor for acquiring the fold practically under which it can bind actually tetracycline. There are certain uses in such a ribose switch, but I just want to skip ahead and come to our experiment. So now the question is, you can see here the binding pocket, and these are the only methyl groups in this complex. So RNA on its own does not contain any methyl groups. So we can control with this binding practically of the small molecule the molecular dynamics within this complex. And now the question is, can we hyperpolarize more than 500 carbons in the RNA by only those 3 methyl groups in tetracycline? And the answer is clearly yes, it works beautifully. This effect is very efficient. We see a uniform hyperpolarization of the RNA under complex binding conditions. What we have also done is we have made a control experiment where we, the only thing that we changed is we did not add any magnesium 2+, and where we made sure that there's no divalent metal ion ever in contact practically with our RNA. And under those conditions, we do not see any spectral contribution from the RNA, even though the RNA is in the same concentration in this solution than here. The only, and tetracycline is also there, the only difference between those two samples is here. We have a bound complex between the two constituents. Here we don't. There are two separate molecules practically swimming around or being frozen practically in the solution without having any dipolar contact. And this is one of the very few examples where we can introduce complex specificity by such an experiment. So that means that we can measure the NMR signal of a bound complex population, even though there might be a population of unbound constituents without giving any interference of the spectrum. We have shown actually that this works also in mixtures. Here we have a competition assay between a binding RNA, a binding aptamer, and a mutant with a much smaller affinity. And here we see that if the binding wild type RNA practically is present, we only see the spectral contribution of this and not of the non-binding partner. I will skip over this. Sorry for the messy or busy slide. What we also can do is we can use this type of spectroscopy to support practically finding the most likely tautomer of tetracycline, which is finally bound in the RNA by using this complex selective spectroscopy. All right, for the last few minutes, I want to show you one final example of this method, and this is on a so-called L7AE RNA subunit of the BOX-CD-RNP complex. This is part of a larger enzyme which performs 2'O methylation of ribose in ribosomal RNA. And here you can see the binding interface or the interaction interface between the protein and the RNA in this complex. And what we've done here, we have further refined our isotope labeling strategy by using carbon-13 or uniform carbon-13 labeling on the adenosine in the RNA. However, in the protein, we have only labeled the methyl groups of isoleucine, leucine, and valine using Lewis case labeling strategy. So that this means that all the other carbons practically in the protein are in natural isotope abundance. Only the methyl groups of isoleucine, leucine, and valine are labeled. And with this, we can again here look at our so-called stream DNP spectra, more specifically the dynamic buildup of the polarization on the carbon nuclei. And you can clearly see that first, the methyl groups, you can see here, they build up rather quickly. After about 16 seconds, they are fully built up. The carbons of the RNA take longer to build up, and this is because once the polarization has been built up practically on the methyl carbons, it takes more time to be transferred to the carbons of the RNA by dipolar interactions or spin diffusion. So now I will just flip those around, so it's easier to compare practically specific and unspecific spectra. So this is just the inversion practically of the spectra mathematically. And now we can compare practically just a cross polarization spectrum. This is done by proton to carbon Hartmann-Hahn cross polarization. This delta dp sub, which is our screen DNP spectrum at a build-up time of 16 seconds. And here a DAR spectrum, which is just a spin diffusion mixed spectrum practically, where we can see the context here between one methyl group and the different RNA carbons of one nucleotide of adenosine 19, and this is the methyl group is isoleucine 93 delta. There's a second minor contact here between valin 95 gamma 1 and adenosine 18. You can see here much weaker signals. And now the interesting thing is that in this cross-polarization spectrum, we see practically all nucleotides unspecifically. However, in our screen DNP spectrum, we see that the peaks here, or most of the peaks which are most prominent here, match nicely with the resonances we see here for the specific contact between this methyl group and this adenosine 19 resonances. And this becomes even clearer if we compare practically the screen DNP spectra at different times. And don't have time to go into the details here of our analysis, but we can clearly see that A19, adenosine 19, also here shows preferential buildup, particularly at the shortest buildup periods of only about 4 seconds or so. So this gives us really a tool where we can utilize this specific contact between this isoleucine 93 delta carbon here and this adenosine 19 to really highlight only this part of the RNA. We have also shown that this is indeed the dominant interaction by mutating this isoleucine 93 away. And then we see that practically the whole transfer collapses and there's virtually no transfer left between the protein and the RNA. And what we can also do, but I will go very quickly over this for the sake of time, is that we can use solid-state NMR specific recoupling sequences, for example, R squared, which stands for rotational resonance, where we match our MAS frequency to the spectral separation of 2 spins we want to recouple practically, and this then enhances practically the transfer efficiency, as you can see here, for example, so that we can even enhance specific contacts even further. And we have recently also investigated this more in detail in a model system here, ethyl acetate. And this was the bachelor thesis by Eddie Bensons. And with this, I want to come to the end. I mentioned again Beatrix Süß's contribution for this collaboration with the RNA, the hammerhead, and the tetracycline binding aptamer. And the RNP was done in collaboration with Alexander Mashanka from the Leibniz University in Hanover. And of course, I thank my group and the funding agencies, most importantly, DFG with the Eminöter program. And before I close, I want to take one more minute to announce a conference to be held in Rostock in September this year. And this is the 45th FGMR discussion meeting of the GDCH, so the Gesellschaft Deutscher Chemiker subdivision magnetic resonance. And this will be in jointly with the Danish, Swedish, and Polish magnetic resonance communities. And I hope we will have I'm sure we will have a vibrant meeting here in Rostock, and would be happy to see many of you there also. And with this, I want to thank you for your attention.